Welcome to VJOncology podcasts, where we deliver clinically relevant insights from the forefront of oncology research and practice. In today's episode, we focus on precision oncology and advanced non-small cell lung cancer. We'll hear from 4 leading experts on unmet needs in rarer driver alterations, the latest data across KRAS G12C, HER2, and EGFR exon 20 insertion mutated disease, how clinicians are navigating an increasingly crowded first-line EGFR space, and what the emerging data on bispecific T-cell engagers mean for the future of immunotherapy in thoracic oncology. We begin with Mariana Brandau, who discusses the unmet needs in BRAF non-V600E mutated non-small cell lung cancer and shares findings from a global physician survey on treatment decision-making in the evolving first-line EGFR mutated landscape, including the growing complexity introduced by osimertinib-based combinations.
This is a complex topic because indeed these are a rare population of patients. And when we have mutations in BRAF, usually they are classified into 3 different classes according to the alterations in the protein and also the protein function according to these different mutations. So I have the class 1, which is the V600, namely the V600E, which is the one that we know the most from melanoma and other solid tumors, but we also have V600A, K, D, and in general, they are sensitive to first generation BRAF inhibitors that now we also combine with the MEK inhibitors, so dabrafenib, trametinib, or encorafenib plus binimetinib. And why are they effective is because these class 1 mutations lead to an activation of the BRAF protein, and she becomes active as a monomer. So all alone, very active in the cytosol and being active and activating all the MAP kinase pathway until the nucleus to lead to cell proliferation and survival. And she's always doing that in an independent way from the RAS signaling, which is upstream. On the other hand, when we have a class 2 mutation, what happens is that these RAF proteins also become active, but they have to form dimers or couples. So they have to be together to activate the downstream pathway, and they do it also in a RAS independent manner. But because they are a dimer, if you just give a first generation BRAF inhibitor, that won't work. Why? Because the BRAF inhibitor connects to one of the partners of the couple, let's say like this, and this leads to a conformational alteration, so a change in the structure of the second partner of the couple, and makes it resistant to the BRAF inhibitor, which means that it actually becomes active and leads to this activation of the downstream pathway. So that's what I call the RAF paradoxal inhibition. And finally, we have the class 3 mutations that what they do is actually they'll lead to a low active or an inactive kinase protein, that what happens is that this BRAF protein, she tights in a better, like more tight, we have to repeat this. So this BRAF couples itself to RAS in a tighter way. And so it amplifies the signals that come from the RAS activation to the downstream pathway. And that's why it doesn't work to inhibit that's BRAF, also because they form dimers. And so it's the same thing from the class 2. So it's important to put a bit of context. So this survey was distributed last year between January and September. And last year we had tons of data coming up on that space. So we have the data from the overall survival of the Mariposa study. So first line amivantamab plus lazertinib versus osimertinib. And later on that year, so after the survey had already ended, we had the overall survival data from the FLORA-2 trial. So combining chemotherapy plus osimertinib and both versus osimertinib. So both trials showed a similar benefit in terms of overall survival. But what we see in the survey that we have is that overall, physicians think that they will give first line single agent osimertinib to around 60% of their patients. Then chemo-osi to around 30% of their patients, and amilase to around 10% of their patients. For me, this was surprising indeed, given the data that we have nowadays. But I think that now that if we would repeat the survey now, probably the chemo-osi or the amilase combinations would be much more used. But what we saw, and that was what we're expecting, is that physicians would prefer to give osi monotherapy to more senior patients, more frail patients, patients with comorbidities, patients that value quality of life over duration of survival. And that's what we know from most clinicians do. They have this discussion with their patients in their clinics. There are patients that, of course, will not even discuss chemotherapy because they have comorbidities or other impediments. Then amivantamab-based, we might, but there is also an important education that has to be done, and the patient has to be really following all these prophylactic measures to decrease rash. So I think that it was a nice survey to understand how the treatment landscape is evolving in the real world. So like how physicians are putting all of this knowledge into their clinical practice.
Next, Adrian Sacher provides updates across 3 areas of active interest to ELCC 2026, including the Candlelit 001 and Be My Own Lung 1 study, as well as final overall survival results from the Become Phase 2 trial.
The Candlelit 001 study was a multi-cohort Phase 1 study looking at a novel KRAS G12C off inhibitor, calderasib, also known as MK-1084. In the study, we looked at calderasib either alone or in combination in both previously treated metastatic non-small cell lung cancer with a KRAS G12C mutation, as well as in treatment-naive patients, looking at those combinations. In terms of findings from the study, we were able to demonstrate that calderasib has a clinically significant but modest response rate in previously treated patients, but that those responses are fairly durable with a median duration of response of 22 months. When we looked at the combination, so calderasib with pembrolizumab or calderasib with pembrolizumab and chemotherapy, we also saw clinically significant responses and potentially durable responses as well, with a median progression-free survival in the calderasib and pembrolizumab treated patients of almost 29 months. And perhaps most impressively, amongst PD-L1 high patients, we saw a response rate of 87%. So the vast majority of people had a great radiographic response to this treatment, and that was also durable. We actually haven't yet reached the median progression-free survival for that subset. Generally, these combinations were tolerable, and the rate of the side effect that we worry the most about with KRAS G12C inhibitors, which is autoimmune hepatitis, was relatively modest. So I think this combination is very promising to look at in randomized studies, and those are already ongoing. This was a study looking at a novel tyrosine kinase inhibitor, zongertinib. The study looked at treatment-naive patients as well as patients with brain metastases. It was very fascinating in that we saw that the response rate for zongertinib was relatively high at 76%, so it's a very compelling finding. And in addition, we also saw a fairly high rate of responses in patients with brain metastasis within the CNS. I think taken together, this is, I think, a very good example of a targeted therapy that is both active generally, it's active in the CNS, it's well tolerated. And I think from both our HER2 as well as our EGFR exon 20 patients, we're seeing more and more of these novel targeted therapies that are quite active. So the field is very promising, but it is increasingly crowded. So the Become Phase 2 study was evaluating a very interesting strategy as becotarib, which is an EGFR antibody combined with high-dose osimertinib in EGFR exon 20 mutated metastatic non-small cell lung cancer. The study demonstrated good activity overall, so we were seeing real clinically significant responses across the different mutational subsets. The study also reported overall survival results of approximately 18 months. I think those data are compelling and hypothesis generating, but it's always difficult to interpret overall survival data in a single arm study. I would say generally it looks like this combination is quite active, and it'll be very interesting to see how it evolves over the coming months and years.
Nicolas Girard then addresses the crowded first-line EGFR space, discussing updated PALOMA-2 results and how tumour boards are navigating between osimertinib monotherapy, osimertinib combinations, and amivantamab-based regimens without head-to-head data. He also examines competing neoadjuvant and perioperative immunotherapy strategies in resectable non-small cell lung cancer.
Well, this is first-line treatment for metastatic EGFR mutated non-small cell lung cancer. We have the PALOMA trial with IV amivantamab plus lazertinib. At the time of the Mariposa trial, actually the safety management for amivantamab lazertinib was unclear. So then many studies have been conducted to optimize this management. SKIPPER for infusion-related reactions, Cocoon for the dermatologic side effects, VT prophylaxis with anticoagulation. And along with that, amivantamab moved from IV administration to subcutaneous administration. And in PALOMA-2, there are several cohorts, including a first-line cohort with subcutaneous amivantamab every 2 weeks, plus lazertinib. So these are the data that I presented at the ELCC 2026 meeting, implementing prophylactic anticoagulation, not dermatologic prophylaxis, but investigators had some awareness about the side effects. And with a subcutaneous formulation, we have a lower rate of administration-related reactions. So quite interesting to see that we have an optimization of the administration of amivantamab, probably leading to a better exposure of the patients, less dose interruption, less dose reduction. And at the end, we have a quite high response rate, 85%, so quite similar to Mariposa, but the PFS is actually higher. It was something like 24 months in Mariposa. Here it's more like 28 months. So maybe it's related to the selection of patients, we don't know, but maybe it's related to the subcutaneous and some of the prophylactic measures. So there are still some ongoing studies like the Copernicus, integrating sub-Q, integrating dermatologic prophylactic management and VT prophylaxis to optimize the management of patients receiving this combination. That's a very good question. Probably looking at the data, there are some differences. Obviously, chemo-free versus targeted therapy. Also, the subgroups. We see that in FLORA2, the magnitude of benefit with adding chemotherapy is observed in patients with aggressive disease, CNS disease, multiple metastases, ctDNA positive. But in Mariposa, the same benefit is observed not only in those patients with aggressive disease, but also in patients without such aggressive disease. So probably a wider benefit with Mariposa as compared to FLORA2. Then obviously the safety profile of these strategies is completely different, but with FLORA2 you have the chemotherapy-related side effects. With Mariposa, it's more like the cutaneous side effects that are prominent. So you have to engage the patient with the understanding of the prophylactic treatment for these side effects. Finally, the sequencing is also an issue, even if the first-line treatment is a major component in the survival of the patients. How to sequence? We have seen during the meeting some very nice data around re-challenge of chemotherapy after FLORA2. So with some high variability in the chemo-free interval and the duration, the actual efficacy of a re-challenge. So how to integrate re-challenge, knowing that most of the second-line options have been developed post-osimertinib in combination strategies such as Mariposa 2, Harmony A with ivonescimab or others. So there is a lot probably to understand a little bit more around those questions. Are pushing for neoadjuvant, pure neoadjuvant, because the figures for survival, PCR are actually similar to that of perioperative as of today, but probably we don't know right now who are the patients benefiting from perioperative versus only neoadjuvant. Probably patients with complete pathological response do not need to further treatment, but again, there is some heterogeneity there. Maybe biomarkers, as we discussed during the session, ctDNA may be of interest to track the patients with a higher risk of recurrence. It's also a matter probably of what is the depth of the pathological response, kind of a continuum from complete pathological response to patients with residual disease. There are some trials ongoing, so it will be very interesting to look at these and enroll patients in such studies.
Finally, May-Lucy Meyer reviews the available data on bispecific T-cell engagers in non-small cell lung cancer and mesothelioma, how they compare to existing immunotherapy approaches, and what clinicians need to understand about their safety profile ahead of broader clinical adaptation.
It's much more limited in the field of non-small cell lung cancer and mesothelioma as compared to what we've seen in small cell lung cancer, but there wide variety of targets being explored, mostly in preclinical stages. And clinical stages are more like phase 1 and 2 trials. But we have emerging signals, so I'm confident in the future we might see more of them. So this is a very good point, and probably we will learn also by using them progressively. But we know that it's very important to have a good baseline assessment of the patient and to know also if we need to use prophylactic drugs. And a key challenge is also the setting where you're using them, because are you in a place where you have a hospital far? So maybe this can be managed in an outpatient way, but if it's not the case, then this patient probably require hospitalization. And now we've seen that we have quite a lot, nearly half of the patients do develop CRS with T-cell engagers, but for the majority it's grade 1 and 2. Which can also be challenging to manage, but it means that these drugs can be used in the clinic, and there are other more difficult to treat toxicities such as ICANS and some other that are rare, but still we still need to learn from those.
Thank you to all of our speakers for their insights from ELCC 2026. Across these 4 segments, we've explored rarer driver alterations, the latest targeted therapy data, first-line EGFR decision-making, and the promise of T-cell engagers in thoracic oncology. For more, visit VJOncology.com and don't forget to subscribe.